So in these two sections, we're going to talk about how scientists actually make these trees and how you can do it yourself. So when scientists are attempting to make a phylogenetic tree, they take information, a lot of information into account. They look at the morphology, so the physical look of the organism. They take a look at the genes, the biochemistry, etc. And then they try to use these similarities to inform phylogenies that result from shared ancestry. But it's important to note that these phylogenies can change as we gather more information from the organism. They're really just hypotheses about how these organisms are evolutionarily related. So looking at their phenotype and their genetic similarities, we talked about in an earlier chapter about homologous structure. So if they have the same ancestor, that's where we get the root homology from, or homo, then they are considered to be related. And then if they have these similar morphologies or DNA that are very similar, then usually scientists will hypothesize that these organisms are more closely related than organisms that have different structures or sequences, which makes sense. So one important thing is that they have to be careful that they make their decisions about relatedness as a result of homology as opposed to analogy. If you remember analogy, we can see organisms that have similar characteristics, but it's because they evolved in a similar location and had to adapt to similar environments. But it doesn't necessarily mean they have a common ancestor. Convergent evolution is usually what causes analogous structures to be seen. And this is when there are environmental pressures and natural selection acting on those pressures that cause there to be similar adaptations in different organisms, even though they may not actually be related. So that's like the bird and the bat wing. Both of them evolved wings because they both had to evolve to fly, but they are not actually as closely related as other organisms are. So as forelimbs, bat and bird wings are homologous in terms of what their function is and how they evolved, but actually working as a wing, they're analogous structures. And so we have to kind of pay attention by comparing fossil evidence and how complex these structures are in order to determine whether they're homologous or analogous structures. Typically, if these two similar structures are very complex, that indicates they're probably homologous, because if you think of it, the chance that two very complex structures would evolve independently is a lot less believable than the fact that you would get one complex evolution and then different branches from a common ancestor. Molecular homologies are based on the DNA sequence of nucleotides between different taxa, and so this is something that is really new into the field of systematics. We didn't have this ability before to evaluate molecular homology, but now we can by using computer programs. And initially this can be very challenging because, as you can imagine, a simple deletion might cause there to be a shift in the reading frame or an insertion, for example. However, in these computer programs, scientists are kind of able to line up DNA sequences and see if they can determine where insertions and deletions have happened, thereby, as you can see, they've kind of now lined them up, being able to see, oh, there really aren't that many differences between this organism and this organism. It just so happened that because of insertions and deletions, we saw a shift in the frame, but really their nucleotide sequence was very similar. You can see down there the CCAT, the CAs match up, and they're very closely related. So if we see shared bases in nucleotide sequences that are otherwise very dissimilar, those are called molecular homoplasties. We don't pay as much attention to them or give them a lot of validity, but it is important to kind of distinguish what's a homoplasy versus what is a legitimate relation. Then we can talk about how they actually build these trees. So once they identify homologous structures, the systematists can use them to infer what a phylogeny could potentially look like between organisms. And this process is known as cladistics. So this is how we build phylogenetic trees. We use cladistics, which is the classification of organisms that are related by a common ancestor. And a clade is a group of species that includes an ancestral species and all of its descendants. So we can have clades nested within each other, but not all groups of organisms are necessarily classified as a clade. So we'll take a look at some of those. The valid clade is a monophyletic group. So mono meaning one. This is a group that consists of an ancestor species and all of its descendants. So if you take a look at this picture, Group 1 represents a monophyletic group because we have the ancestral species, which is whatever species this was, and then all of its descendants. Group taxa A, taxa B, and taxa C. You can see their most common ancestor was this one singular organism. A paraphyletic group can consist of an ancestral species and some, but not necessarily all of the descendants. So in this case, you can see group 2, we have an ancestral species. Group 2 is the one that's shaded, so D, E, and F taxa. 2's common ancestors by this branching would be D and E from over here, and F and G. But you can see this group is paraphyletic because it only is containing some of the ancestors. And we don't know why in this case they're grouping them in this way, but you can see that. If the shading included the G, then it would be monophyletic. But because it doesn't, this is a paraphyletic group. And then finally, a polyphyletic group, poly meaning many, can consist of various taxa with different ancestors. So if we take a look here, we can see all of these different groups, all these different taxas in group 3 have various common ancestors, but not necessarily the most recent. So we see A, B, and C and D are all taxa that ultimately are related to this 3, but their most recent common ancestor is not group 3. So that's why they're not monophyletic. And here you can see them all compared. In a paraphyletic group, the most recent common ancestor of all members of the group is a part of the group. So you can see here the most recent common ancestor is number 2, and they're part of the group. But in a polyphyletic group, the most recent common ancestor is not part of the group. So here, between A through D taxa, the most recent common ancestor between all of them is number 3. But because 3 isn't included in part of the group, that's part of the distinguish between para and polyphyletic. This shows us some different groups here. So we can see this shows us the evolutionary relationship between even-toed ungulates as well as a lot of other creatures. So interestingly enough, even-toed ungulates, they include organisms that have an even number of toes. So we can see here they are actually very closely related to hippos, and this is a paraphyletic group. However, cetaceans and seals are also closely related, but this would be polyphyletic because their most recent common ancestor is all the way back here. So it's not included as part of this group. When we are taking a look at the characters by which we want to judge whether these groups are related, we look at shared characteristics and different characteristics. A shared ancestral character is a character that originated in the ancestor of the taxon. So for example, in mammals, the backbone is a shared ancestral character because the original ancestor had a backbone. A shared derived character is one that is unique to the clade. So this is something that first appeared in that particular clade. And so, for example, hair in mammals is a unique shared derived character. When we are looking at evolutionary relationships, it is helpful to know in which clade a shared derived character first appeared because that can help us identify what the most recent common ancestor was like. So over here, we can see we have a few different groups. We have leopards, turtles, frogs, bassfish, lamprey, and a lancelet. And over here, we are keeping track of what these groups, what characteristics these groups in particular have. And as you can see, based on looking at this chart, we see that except for the lancelet, all of the groups have a backbone of vertebrae. 4 of them have hinged jaws. 3 have forelimbs. 4, like the number 4. 2 have an amnion, that's like the sac that gives nutrients to the child, and only 1 has hair. So in this way, because we can compare which organisms have the most shared similarities, we can determine their evolutionary relationship. So turtles and leopards are more closely related, but this is where the change happened. Then frogs are pretty closely related to turtles and leopards, but this change happened, the appearance of the amnion. 4 limbs, the growth of limbs is a change that happened in between the bass or bass fish, frog, turtle, and leopard, and so on and so on. So you can see how they're more distantly related as we move outward on the tree. An outgroup is a species or a group of species that is closely related to the in-group, which is the species we're studying, but the outgroup has diverged before the in-group. So typically, they don't share any similarities with the group that's being compared. They're kind of as close as you can get without having any similarities. Any characters that are shared by the outgroup and the in-group, if there are any similarities, are known as ancestral characters, and so these are traits that were present before the groups diverged between their common or from their common ancestor. Also, typically the length of the branches on the phylogenetic trees doesn't necessarily matter, but in some cases, you'll be told if this is the case, the length of the branch can reflect the amount of change that's taken place in a particular DNA sequence of that lineage. So, for example, when we see these longer branch lengths, we can know that might mean that there's been more genetic changes between a fly and a mouse than, say, a human and a mouse. So we're much more closely related to a mouse than a fly, if that makes sense, based on these lengths. And hopefully, you will also get to experience some trees where branch length can represent the actual amount of time it took for those changes to occur, and that we determine from looking at the fossil record. So similarly here, we can see the fly evolved from the most recent common ancestor about 500 million years ago, whereas humans only about maybe 50 to 75 million years ago. The last thing is about phylogenetic trees is that our goal in the end is to achieve what's known as maximum parsimony. So systematists can never be 100% sure that they have found the true phylogenetic tree, but they use the principle of parsimony to determine what is the best option. This basically means that the tree that requires the fewest evolutionary events, aka the fewest appearances of shared derived characters, is the most likely. And this is because, kind of similar to one of the past examples, the idea is that we probably got here by the simplest methods possible, aka the fewest number of changes, as opposed to everything evolving differently. And computers have greatly helped us out with this. So in class, I'm going to walk you through this example, but I will go ahead and leave this here. I'll take some time so you can pause and take some notes. This is figure 20.15 in your textbook, but notice between these 3 hypotheses of how these species might be related, ultimately what you're going to see, these are the changes in nucleotides from the ancestral sequence. For this tree, it shows 6 different events happening to get there. These 2 trees, it takes 7 changes, 7 nucleotide changes to get there. So this tree is the most parsimonious because it requires the least amount of effort to make. Finally, once again, it's important to note that these phylogenetic trees are simply hypotheses. We don't know for sure how the species are related. We continue to add and adjust and change our trees based on which data we have, so whether we find more morphological data, more molecular data, or more fossil data, and then we modify these trees as we find new evidence and new information. So we are able to kind of determine how these creatures are related